I'm Dr. Neil Kalman, Chair of Family Medicine and Community Health at Sinai and President of the Institute for Family Health. And it is a profound pleasure this morning to introduce Dr. Florian Krammer, who graduated from the University of Natural Resources and Life Sciences in Vienna, Austria in 2010. He's one of the truly brilliant minds at Mount Sinai that we rarely come in contact with in our work, but we're so excited about having him here today. He actually received his postdoctoral training in the lab of Dr. Peter Police at the Icahn School of Medicine, working on hemagglutinin, stock-based immunity, and universal influenza virus vaccines, for which I believe he got one of the largest grants ever given by the NIH. In 2014, Dr. Krammer became an independent principal investigator and is currently Mount Sinai Professor of Vaccinology at the School of Medicine. His work focuses on understanding the mechanisms of interactions between antibodies and viral surface glycoproteins, and on translating this work into novel, broadly protective vaccines and therapeutics. The main target is influenza virus, but he's also working on coronaviruses, flaviviruses, hantaviruses, filoviruses, and something I never heard of called arenaviruses. He's published more than 240 papers on these topics, and since 2019, he served as a principal investigator of the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, which develops improved seasonal and universal influenza virus vaccines that induce long-lasting protection against the drifting seasonal zoonotic and future pandemic influenza viruses. So we asked Dr. Krammer here to tell us the inside scoop on what's going on. He's going to focus mostly on the coronavirus story, and it's my pleasure to turn it over to you. Thanks so much, Florian.
Thank you very much for this nice introduction. Let me try to share my screen. Too many windows open, of course. Okay, I hope you can see my screen. All right, so what I'm going to try to do is I'll start a little bit with discussion about immunity to SARS coronavirus 2 and also some work that we did here at Mount Sinai, and then I'll go into vaccines, and then I hope I can answer a lot of the questions that you might have about vaccines that are currently used in the population and vaccines that are coming in the next few months. Just start with my disclosures. We are working here on SARS-CoV-2 vaccines. I'm not going to talk about those vaccines today because they're in early stage of development, and we're also working on antibody tests for SARS coronavirus 2. So let me just start with immunity. You probably are very familiar with schematics like this by now. This is a schematic of a coronavirus, of SARS coronavirus 2. We have an RNA genome on the inside that's covered by a nucleoprotein to protect the RNA. Then there's a host cell-derived envelope that has two proteins, the envelope protein, which is an ion channel, and the matrix protein. And then we have this humongous spike protein on the surface. And this protein is very important because it actually is what the virus uses to bind to our cells, to the receptor that it is using, ACE2. And here on the right side, I'm showing a crystal structure of that spike. This is a homotrimeric protein, so you have three units that come together to form this spike, and one of these units is shown here in blue, and its receptor binding domain is shown in red. So this little red part here is what actually docks to our cells. And why I'm stressing the spike so much, of course, if you can imagine if an antibody binds here in this area, the virus cannot dock to our cells anymore, and the virus is neutralized. And that's, of course, a very important concept. So what do we know about immune responses to the virus? We get strong antibody responses to the nucleoprotein. They might not be beneficial because the nucleoprotein is inside. These antibodies are usually not neutralizing, but they're a good diagnostic tool to see if somebody had been infected. And then we get also good antibody responses against the spike protein, and we know that these can be strongly neutralizing. We also get cellular immune responses against the virus, and that includes CD8 and CD4 T cell responses. We see that the CD4 response is a little bit better than the CD8 response, and it's distributed throughout the viral proteome. So you get good T cell responses against the spike, but also the nuclear protein and other proteins. So a little bit about what we did here at Mount Sinai. We got the sequence of the virus on January 10th, so basically 11 days after the first cases were reported, and we immediately started to work on reagents and assays to measure antibody responses. And relatively quickly, we started to talk to the clinical laboratory here, in pathology, to Carlos Cordon, and we transferred the assay to their clinical laboratory, and they started to screen for people who already had antibodies because they wanted to start a plasma program so that they can treat patients with convalescent plasma. And so when we had the first 1,300 individuals screened, we looked at the antibody responses that we found, and we had two types of donors: PCR confirmed donors, so people who actually we knew had an infection with SARS-CoV-2, and the vast majority of them actually made antibodies to the spike protein. And then we had COVID-19 suspected donors. Remember, this was early in 2020. The flu season was still going on. A lot of people had infections and then said, oh, it's probably COVID. And so what we found here was that only 40% of these suspected donors, which were never PCR confirmed, had antibodies. About 60% did not. They probably had other infections that were prevalent still at this point in time. And then we went on over time, sampled a lot of people. As you know, Mount Sinai opened antibody testing to all employees. And so many samples were tested over time. I think we have now tested in the clinical lab more than 100,000 samples. And on the left side, I'm just showing the antibody titers that are measured in positives. This is from 30,000 positive individuals. And what we see here is they stratified this into different types of titers. 1 to 80 and 1 to 160 are low titers, and very few people seem to have low titers. 1 to 320 is an intermediate titer, and about 20% of people get these intermediate titers after infection. And then you have 1 to 960 and 1 to 2880 or higher. And that's 70% of all people who are positive, and that's considered high titers. I know that this scale recently changed. So if you get tested now, you get units per mil, but there's some kind of conversion that they offer. So if you have an old antibody test and the new one, you can kind of calculate the difference. And here on the right side, I'm just showing samples that were tested over time. That were positive over time. So this goes from basically April to October. And the blue line here indicates the percentage of people who had 1 to 320 titer or higher, and that oscillates around 90%. The take-home message here is just that when people get infected, they usually mount a relatively robust response to the spike protein. We also wanted to know what these antibodies do. So, you know, when we measure ELISA titers, those are binding antibodies, but you don't necessarily know if they're neutralizing, if they have a function. And so we took a set of 120 individuals and we looked at their neutralizing antibody titers. So this is done in we call that microneutralization assay in a biosafety level 3 laboratory. We measured the neutralizing activity here, and then we plotted that against the ELISA titers, and you see that there's a very nice correlation. The higher your ELISA titer, the higher your neutralizing activity. And this is of course good because that means if we measure ELISA titers, we can extrapolate the neutralizing activity. We also wanted to look at how long-lived these antibody responses are. Remember back in spring, there were a lot of reports about waning immunity, and there was basically one newspaper report that said, yeah, within 8 weeks, your antibodies are gone and will get reinfected over and over and over again. And so we started to look at that. Again, we had 120 individuals from the plasma donor program, and we followed them longitudinally. We took samples from them on around day 30, around day 82, and then 5 months out. We actually have a 7 months time point too, but I don't have that data in the slide set yet. Basically, what you see is that between day 30 and day 82, there's not much movement in terms of the titers. And then there's some drop towards month 5, which is normal antibody response, not very concerning. But of course, the titers do not go away. The antibodies don't go away because that wouldn't make sense biologically. We stratified this also by titer, and what we see here is that people who have initially very high titers have a steeper drop-off, as you can see here, about 3-fold over 5 months. 3-fold is actually not that bad. While people who have initially low titers have very different kinetics. They have an increase from day 30 to day 82, and then a slow decrease again. But what that means is that it doesn't matter if you have initially a high titer or a low titer, the titer will still, or the antibodies will still remain in your body. They're not going to go away. We also looked at neutralizing activity over time, and this is shown here on the left side. As you can see, there's a variety of neutralizing antibody titers in people who had an infection. So there's, all kinds of titers. It's a lot of heterogeneity, but the take-home message basically is that also the neutralizing antibodies don't go away. They go down a little bit, but basically, again, over the course of 5 months, you have about a 3-fold decrease, and the expectation is that they stabilize after 5 months. And even 5 months out, we still saw this nice correlation between neutralizing activity and binding in ELISA. So what does all of this mean? Does this really help us, or these neutralizing antibodies are protecting us? And so in early summer, 2 papers were published by Dan Baruch's group and in rhesus macaques. And rhesus macaques are a pretty good model for SARS-CoV-2. The first study, which is shown here on the left side, is an infection-reinfection study. So initially, they infected these rhesus macaques. This here is RNA copies of the genome copies of the virus in the bowel fluid. So basically, a virus titer indicates how much the virus replicates in the lung. And you can see initially this is high, and then after a few days, the animals start to clear the virus. So it replicates, and then it's cleared. And then they re-challenge these animals. And this is shown here on the right side. And what you can see here is that these animals are protected from re-challenge. You see these blips here of RNA. That's probably the challenge virus, because these animals are intratracheally challenged. So you put a lot of virus into their lungs. And then initially on the first time point, you can detect it, but the virus doesn't replicate. And so that was the first indication that infection is actually protective. Or induces protective immunity. At the same time, they did another study where they used a relatively imperfect DNA vaccine, which gave a variety of neutralizing antibody titers. And when they challenged the animals, they found that the protection of the lung and the neutralizing antibody titers correlated very nicely. So the higher your neutralizing antibody titer, the better your lung was protected, and the better the animals were protected from disease. And so they basically established a correlate of protection in the non-human primate model. And if you look here, the titers are not that high. This is a 1 to 100 titer. This is actually not a high titer. And this was very, very useful information. And as you know, we have correlates of protection for many vaccines and many viruses established already. For example, for hepatitis A, hepatitis B, influenza, where you have the famous 1 to 40 HI titer. If you have that, your risk of getting influenza is reduced by 50%. For measles, we have a microneutralization assay. And this is all very useful because it basically can tell you if an individual is susceptible to infection or not. If somebody is above these antibody levels, they are protected. If they're below, you might want to vaccinate them. But we don't have something like that for SARS-CoV-2. But there's more and more evidence coming up, and I think we'll soon have a correlate of protection, like we have it for many other viruses. The first evidence that neutralizing antibodies may be a correlate of protection in humans, I mean, for monkeys it has been shown, came from a really nice study that was done in Seattle. The Bloom and Graninger labs, they're basically enrolled all the crew of a fishing vessel, a commercial fishing vessel, into a study. So they had 122 individuals on the ship, and they found that 3 of them had already neutralizing antibodies. And then the boat left for their fishing trip, and as you know, when you have an outbreak on one of these boats, specifically on cruise ships, we have seen that the attack rates are very high, and this happened on this fishing boat. They had an outbreak that an attack rate of 82.5%. You know, people interact very closely there. It's close quarters, so these are ideal conditions for the virus. But all 3 individuals who had neutralizing antibodies were protected. They did not get infected. And that's a very small number, but it's actually statistically significant. And so that was the first study that provided some evidence. But now there's really large studies from the UK, mostly in healthcare workers. There was one published in the New England Journal of Medicine, and another one is now on a preprint server, the Siren study. And both of these studies show that infection and having antibodies gives you a very high protection actually against reinfection. And I'm not talking about symptomatic infection. I'm actually talking about protection against any type of infection. So they found that the efficacy, so to say, of having an initial infection and having antibody against reinfection is actually better than what you get from the vaccines. And of course, I'm aware that reinfections may happen. And they also found some reinfections, but if you calculate the efficacy, so to say, or the risk ratio, it looks very good. And so there's a lot of indication now that having antibodies and having had an infection with SARS-CoV-2 is actually protecting you from future infections. But I want to switch gears now to vaccines, since we have talked now about the basis for vaccines, immune responses. I want to go into vaccines and vaccine development a little bit. So one slide that I really like to show is about vaccine development and how long it usually takes. And this is depicting here classical vaccine development, which usually takes about 15 years. I think the fastest vaccine that has ever been developed was mumps, if I remember correctly, and that took 5 years before COVID. And usually you start with a design and exploratory preclinical phase. This is usually done in an academic lab. It's basically going back and forth in an iterative approach, trying to optimize your vaccine, vaccinating a lot of mice, unfortunately killing a lot of mice because they're not protected, going back, redesigning, and this can take 4 years. If you have something that's promising in the lab, you usually need a commercial partner. You partner up with a company, and then you start to develop a process, how to make the vaccine. That takes a long time. You do more preclinical work, you do toxicology studies, and that usually takes 2 to 4 years. And you also need to convince them that they actually want to go forward with a clinical trial and spend all the money. And then at some point the IND is submitted and you start a phase 1 trial, which is usually in less than 100 individuals. And you look at primarily safety and a little bit immunogenicity. That takes about 2 years. If that looks good and the company is still interested and there's funding, so you go into a phase 2 trial. Phase 2 trial is larger and it's more expensive. We're usually talking about $15 million here. Takes another 2 years. You look at different doses and so on and so forth. And once you have the results of the phase 2 trial, you usually sit down and really take a hard look at the data and at the market. Because the phase 3 trial is going to cost between $300 and $500 million. And you really need to be sure that you want to invest that money, because if there's any doubt about the data or about the market, because if there's no market, you or if the market is not big enough, you might not want to spend that money. You need to sit down and decide if you really move forward with that. A lot of vaccines die in phase 2 or after phase 2. And then if the funding is there, you might go into phase 3. You have to predetermine what success is before you start the phase 3. And the phase 3 might take a long time, not necessarily because you have to do a lot of safety follow-up, but because you have to accumulate cases. And if there is few cases, it takes a long time to get enough data to calculate efficacy, statistically significant efficacy. You can circumvent the long time by making the trial bigger, but of course that makes it also more costly. Once you have that data, you submit a biologics license application to the FDA. Usually they sit on it for quite some time. They might have questions. They don't necessarily prioritize one company over another. And then you might be granted a license. And once you have that, you start production and distribution. And this takes a long time. Now for SARS-CoV-2, that was very different. We skipped a lot of stages. For example, the design and exploratory preclinical phase that was not needed. SARS-CoV-2 is very similar to SARS-CoV-1, which was circulating in 2003 and was then eradicated. It's also very similar to MERS coronavirus. Vaccines have been designed for these viruses. We knew exactly how the vaccine has to look and how the antigen has to look. So basically, once the sequence was there, we had vaccine approaches. So this was completely skipped. And then specifically with the vectored vaccines and the RNA vaccines, you don't need process development because you just change the sequence. There's no need to go into that. And that is also the reason why we see now the RNA and the vectored vaccines first on the market. For other vaccine approaches, you need to do some process development. But Moderna, for example, or Pfizer didn't need to do that. They just used the process they had. And that was the reason why Moderna was able to start a phase 1 trial in March. And then what they did was they basically did an adaptive trial design where you have a phase 1. If that looks good in terms of safety, you make it larger and becomes a phase 2 trial. This is not something special. We are doing that now all the time, but it saves, of course, time. And once the interim results for phase 2 were available, they started a phase 3 trial at risk because usually you wouldn't do that. You would want to be very sure about the immunogenicity data. But phase 3 trials were already started in the end of July. And because there was so much government funding for this, there was de-risking for the companies, so they could do that. You would never ever do that if you develop a vaccine because there's too much risk involved here. And then they also started to do production at risk, which is another thing that you would never do. And then basically, once phase 3 efficacy data was there and enough safety data had accumulated, biologics license application, in this case for an emergency use authorization, was submitted to the FDA. And of course, the FDA did not just put that in their queue and said, yeah, we'll deal with it once we have time. No, of course, they looked at it right away. And so that sped up things as well. I wanted to stress, though, that in the US, this is an emergency use authorization, but in the European Union, this is a normal license now. They didn't emergency authorize it, they really licensed the Pfizer and the Moderna vaccines already. And so because of all these things, it was possible to develop these vaccines basically within a year. But that doesn't mean that any corners were cut in terms of safety. That's important to stress. Basically, knowledge, technology, and money that made this possible. Okay, so there's a lot of different types of vaccines that are in development right now. I'm just going to show that slide. I'm not going to go through it because most of these vaccine platforms are not really going to be relevant in the near future for us. But I just wanted to show you that there's many approaches, and many of them actually show initial success in animal models and also in early stage clinical trials. What we should focus on are these 4 types of vaccines because they're more relevant for us. There are inactivated virus vaccines for SARS-CoV-2. You make them by taking a clinical isolate, you grow it in a bioreactor on cells, you inactivate the virus, and then you purify it and you use it as vaccine. And this approach is taken right now in China and also in India. There are vaccines that are on the market in some countries globally already, including in Latin America, and then also in India, for example. But in the US, those are not licensed at this moment in time. Then we have recombinant protein vaccines. There's actually one that is coming in the US, produced by Novavax. Those are classical vaccine technologies. There's an influenza vaccine that's based on recombinant proteins on the market. The hepatitis B vaccine is similar, and the HPV vaccine is also made in a similar way, so nothing new. Then we have viral vectors, and this is some newer technology that has been used in licensed vaccines. So there's 2 Ebola vaccines that are licensed in Europe that are based on viral vectors. But what you do here is you basically take a harmless virus, in this case usually an adenovirus, you remove parts of its genome so that it can't replicate anymore. You give it the information for the SARS coronavirus 2 spike, and then you use that, it goes into your cells, provides the cells with the information for the spike. The spike is made by your cells, you get an immune response, but the virus itself can't replicate. So it's very safe. Again, it has been approved for some vaccines already. And the RNA vaccines work in a similar way. You basically make in a bioreactor RNA that codes for the spike protein of SARS coronavirus 2. So this is a messenger RNA, similar to the messenger RNA that we have in our cells. You basically package that in a fat particle. We call that lipid nanoparticle. You inject that, and then cells take it up, and the ribosomes in those cells recognize the RNA and make that protein on the cell surface, and you get an immune response. So they're relatively simple approaches, and they work relatively well. So just a little bit about the data in animals, and I'm not going to go through this in detail. I'm just going to give you the summary. Basically, a lot of animal studies were done with the inactivated vaccines, the vectored vaccines, RNA vaccines, recombinant protein vaccines. Basically, they all work. You can protect non-human primates from disease and from virus replication in the lung with all these vaccines. What came through during these vaccine trials in animals is that you typically don't protect the upper respiratory tract that well. So there's usually still some replication in the upper respiratory tract, even if the animals are vaccinated. However, if you compare that to non-vaccinated animals, you see that the replication is, there's less virus and for a shorter period of time. But I think it's safe to say that you can't completely protect these animals from infection with vaccination. You can protect them nicely from disease, but there's some replication that's still occurring. And then from the early clinical trials, we saw that the inactivated vaccines do an okay job in inducing neutralizing antibody titers when given 2 or 3 times. For the vectored vaccines, it's more or less a mixed bag. The AstraZeneca vaccine worked relatively okay. The neutralizing titers are not sky high, but they're certainly there. There was early data from CanSino. This is an Ad5 vector that was developed in China. That was not doing that great. They only had very low neutralizing activity. For the RNA vaccines, and again, these are given twice, we saw that after the first vaccination, you don't really get neutralizing antibodies, but after the second vaccination, you get a lot of neutralizing antibodies. And the same is true for the recombinant protein vaccines that are also given twice, where you initially get relatively low antibody titers, and then after you get the second dose, you really make a lot of these antibodies. And that's something that we very often see with vaccines. Just to sum up what's around right now, what's in phase 3 and what's already authorized. We have the 2 RNA vaccines, one produced by Moderna, one by Pfizer. They have shown efficacy, 94% for Moderna, 95% for Pfizer. They are used in the US since December. The AstraZeneca vaccine, it's a mixed bag depending on the study that you look. The efficacy is between 62 and 90%. This is already licensed in the European Union and in the UK. Then we have Janssen or J&J. This is also a vectored vaccine that's only given twice. And this really stands out because of that. That had an efficacy in the US of 72%, which is not bad for a vaccine that you only give once. The Novavax vaccine, which was trialed in the UK and is right now trialed in the US as well, gave an efficacy between 89 and 96%, so it's very good. And then here in red, we have vaccines that have interim results but are not available in the US. One is the Sputnik 5 vaccine that's was developed by Gamaleya Institute, and then we have several inactivated vaccines that are produced by Chinese or Indian companies. They show 50 to 90% efficacy, and then we have another vectored vaccine that is produced by CanSino in China. There's no good data for that vaccine yet, but it's also widely used in some populations. Just to review how these clinical trials are done, typically in a phase 3 trial, you have two groups, a vaccine group and a placebo control group. The vaccine group gets the vaccine. The placebo control group usually gets an injection with saline solution. Of course, you don't tell these people what they got. They're blinded. And importantly, these trials are usually conducted by independent medical centers, not by the companies themselves. And Mount Sinai is part of several of these clinical trials. Then you have an independent committee that watches over the data, which is important for safety, but it's also important for data analysis. And of course, before you start, you have preset time points and success criteria. And then you vaccinate these people and you follow them and you look who is getting COVID-19. And you count in the end the cases in the vaccine group and you count the cases in the placebo control group. And that allows you to calculate the efficacy. And if the vaccine is good, you have a lot of cases in the placebo control group, but no cases or very few cases in the vaccine group. Now, just to review the results of the vaccines that are on the market right now, Pfizer had 43,000 individuals in their study. They had 170 COVID-19 cases, 162 in the placebo group, and 8 in the vaccine group. And since there was a 50-50 split between vaccine and placebo group in terms of individuals in the study, that already tells you that the vaccine is working. They calculated the 95% efficacy overall, and they were able to analyze a subset of 65 to 85-year-olds. They found efficacy of 94% there. This is important because this is, of course, a group that is at high risk, and the vaccine works there. And we're always worried about vaccines in older people because they usually don't respond that well. And they had no significant safety concerns, and the vaccine is now in different degrees of approval in many, many countries. I just wanted to show this slide because it really demonstrates how these results look and how they look like for a good vaccine. Here on the x-axis, we have time after the first dose. The first dose was given here on day 0, the second dose on day 21. And here we have basically accumulation of SARS-CoV-2 cases in the groups. The red group is the placebo group. You see there is a constant increase in cases. And the blue group is the vaccine group. And you see for the first 10 days, they go in parallel, basically. And then there's a deviation of the blue line here. And even after the first group, basically that indicates that there's already protection. And this gets better after the second vaccination. And you see that then there's barely any cases. You still have some cases, and that's normal because as for all vaccines, we're talking about a risk reduction, not absolute protection. But this data is, of course, very good. And I think when people first saw it in the field, they were very happy to see this. Moderna had very similar data. They tested, they did their phase 3 in 30,000 individuals. They had 196 COVID cases, 185 in the placebo group, 11 in the vaccinated group. They calculated 94.1% efficacy, and again, no significant safety concerns, some indication of reduction of asymptomatic infections, and I will get into that. And again, this vaccine is now approved in many different countries. I know people are concerned about RNA vaccines because it's a new technology. I just wanted to stress that this is not the first time we're using this in humans. There have been trials for infectious disease vaccines with mRNA starting in 2013. It's not huge numbers, but of course there was safety testing and immunogenicity testing, and actually Moderna is moving, or was moving, one CMV vaccine into phase 2 because it looked promising. That trial is currently ongoing. So it's not that it's the first time this vaccine is used. There's, of course, a lot of experience from clinical trials already. Just in terms of other vaccines, I already mentioned AstraZeneca. This is a vectored vaccine that is licensed in the UK and in the European Union. The efficacy varies depending on the study. And I'm still not very happy with their data and how they communicate it, and it's a little bit confusing. I think the only thing we can say about this vaccine is that it works, but it's not really clear how well it works. There are a lot of these inactivated vaccines that have been trialed, and there's a variety of efficacies that you get. Sinopharm, for example, at 86%, and they are licensed in some countries. I already mentioned the Gamaleya vaccine. Sputnik 5 seems to be efficacious, but the data is not available. They recently published a report in the Lancet, but the underlying data, as we see it for Moderna and Pfizer, unfortunately not publicly available. Novavax reported 90 to 96% for their vaccine in the UK, which is a recombinant protein vaccine, 50% in South Africa, and I'll get to that. And the J&J vaccine had 72% efficacy in the US, but lower in South Africa, and I'll get to that as well. This is a one-shot vaccine. A little bit about safety. The first thing I wanted to mention is reactogenicity. Reactogenicity is basically a reaction to the vaccination, reaction of your immune system that is very often triggered by release of interferon. So it's innate immune responses, basically. Reactogenicity is usually unpleasant, but really not dangerous. And what we see very often here is injection site pain, headache, fatigue, elevated temperature, myalgia, mild flu-like symptoms, things that are really triggered by interferon. You see these things for other vaccines as well. Some people react badly to flu vaccines with symptoms like that, but I have to stress that they're more common for adenovirus vaccines and mRNA vaccines. And specifically for the mRNA vaccines, we see that the reactogenicity after the first dose is relatively low, but after the second dose it increases in frequency, not necessarily in severity, but many people experience headache or fatigue or elevated temperature after the second shot of RNA vaccine. Again, this is not dangerous, but something to keep in mind because people might not feel well after they get their second shot. This is very transient. It usually goes away after a day or two. The more severe side effect that we are seeing, and it's very rare, is allergic reactions or severe allergic reactions. People who were known to have severe allergic reactions to vaccines were excluded from the phase 3 trials, but when the vaccine was rolled out, it was found that in some people it led to severe allergic reactions, and those are usually people who have a history of severe allergic reactions, and we have now rates for that, and it's approximately 11 severe allergic reactions per 1 million vaccinated individuals for the Pfizer vaccine, and 2.5 per 1 million for the Moderna vaccine. So these are low rates, but certainly something to be aware of if you're vaccinating individuals. And this can be dealt with, of course, with anti-allergic medications. And again, it's relatively rare. Then what about vaccines and pregnancy? This is a little bit of an interesting topic because the FDA or the CDC leave it up to the person who is pregnant to make the choice and find out if they want to get vaccinated or not. But in general, the recommendation is if you are at increased risk of COVID for example, if you're a healthcare worker and you're pregnant, you should look at your risk and you should have a conversation with your physician and then find out and make a decision if you want to get vaccinated or not. So they don't really give good advice for this. They you know, basically leave it up to the individuals who are affected to make the choice. But I looked at the clinical trial data just to provide some information. Of course, there's no good data out there, but there are some data from the phase 3 trials. Of course, if you're in a phase 3 trial for a vaccine, you're asked to use contraception, but there are always people who get pregnant. And I just extracted that data from the clinical trial reports that were submitted to the FDA. And we found, they found that in the Pfizer vaccine trial, they had 12 pregnancies in the placebo group and 13 pregnancies in the vaccine group. For the Moderna trial, was also equally distributed with 7 pregnancies in the placebo group, 6 pregnancies in the vaccine group, and it's similar for the AstraZeneca trial. You know, these numbers are not big enough to draw any statistically significant conclusions, but on the other side, they are in a way reassuring for people who are worried about fertility, who are worried about getting vaccinated during pregnancy. Of course, there's no long-term data, so we don't know, you know, once these babies were born or will be born, because some of them, for some of them, the time is too short to know that. We don't know if there's any kind of problems associated with that, but there's no biological reason to assume that. There's also now relatively good data in laboratory animals, and I think they're doing clinical trials now in pregnant women as well to really make sure that that there is no increased risk. But I find these numbers relatively reassuring, although they are small and maybe not statistically significant. Another point that I wanted to mention is that the vaccines seem to be working better in younger people in terms of immunogenicity. So the antibody responses are higher in younger people than in older people. That has been shown in several of these trials, but also in of course, SARS-CoV-2 vaccines. However, the Pfizer trial showed that even with a little bit of a lower antibody titer, they still get very good efficacy in older age groups. Right now, this is not a problem, but this is something that that we need to keep an eye on, specifically over time, and the titers might go down. So we might see a difference in the loss of efficacy or effectiveness in different age groups. I already mentioned protection from asymptomatic infections and what we found in the animal models. This is a really important topic. Typically, if you get a natural infection, you get mucosal immunity, and you also get systemic immunity. So basically your upper respiratory tract and your lower respiratory tract are protected. If you get a vaccine, typically make very good systemic antibody responses that protect your lung, but you usually don't have good mucosal immunity in the upper respiratory tract. And that might be an issue and might lead to might allow asymptomatic infections. And there are ways around that, for example, intranasal vaccination or mucosal vaccination in general, but these vaccines are not ready for SARS-CoV-2 yet. And so what do we know about protection from asymptomatic infection? In the Moderna trial, when people came back to get their second shot, they were swabbed, and the data for that is already available. And what was seen there is that there was a 60% reduction in asymptomatic infections. The numbers were low. Again, it's hard to make statistically significant assumptions or conclusions, but there was certainly a trend that there was a reduction in asymptomatic infections, and that might even be better after 2 vaccinations. However, I don't think that all asymptomatic infections will be blocked in people who are vaccinated. They might still occur at a certain rate. However, as we can extrapolate from animal models, it's likely that there's less virus that is shed in for a shorter period of time. There's already some data from Israel showing lower CT values in people who had COVID or had an asymptomatic SARS-CoV-2 infection after the first vaccine as compared to people who were not vaccinated. So there's more and more data coming. saying that vaccination actually has a pretty big impact also on asymptomatic infections. You know, I think it could happen that somebody who is vaccinated still gets infected and then passes on the virus. We can't exclude that. But I think the risk that happens is much lower than with a non-vaccinated person. And over time, we'll see how we can quantify that and how that risk can actually be quantified. I just wanted to mention that this is similar to other vaccines like flu or pertussis that are also mostly protecting from disease, but not necessarily from asymptomatic infection. How long does protection last? That's a good question. Likely for years, based on what we know in general about immune responses to vaccines and to SARS coronavirus 2. There are, of course, only limited data because we just started vaccinating people. Moderna had published data over 4 months, and that looks good, but of course, that should look good. So we need to follow people and see how the titers develop over time. And we'll find out if we need to give booster doses at some point in time. That would not be a big problem. We do that for other vaccines as well. But my expectation is that except for troubles with variants, we'll get relatively long-lasting immunity from these vaccines.
Krammer, just to let you know, we're at 8:46.
Okay. I should be able to wrap up quickly. One important question that came up quite often was, should individuals who already had SARS-CoV-2 get vaccinated? And if yes, how often? So the recommendation right now is that people who had SARS-CoV-2 should also get vaccinated twice. But immunologically, that doesn't make sense because they're already primed, and their first dose of vaccine would be like the booster dose for people who had not had COVID before. And we have also heard a lot about people who had COVID and they get vaccinated and complain about stronger side effects. And so with a colleague of mine, with Viviana Simon, we set out to look at that. And what we found is that people who had, and these are these orange balls here, people who had already antibodies to SARS-CoV-2 reacted very quickly and very strongly to the first vaccine dose and developed antibody responses that were as good or even better than the antibody responses in people who were naive initially and then got vaccinated twice. And you know, naive people who get the first shot actually have a slower response and it doesn't go as high. So we believe, and there's now more data from 3 other groups out there that basically show the same, that in people who already had COVID-19, 1 shot might be enough. And we also showed that, yes, indeed, people who had SARS-CoV-2 infection that then get their first shot have more systemic side effects than people who are naive to SARS coronavirus 2. The last topic that I wanted to cover is the variants. You've heard a lot about them. There's basically 3 variants of concern that are around right now. B.1.1.7, this is the British origin variant that has a mutation in the receptor binding domain that makes it more infectious, probably 35% more infectious. That doesn't sound much, but that means that it can take over the whole big proportion of the infections over time. Right now, there's no evidence that it causes more severe disease, and we'll get into vaccines and therapeutics in a second. Then we have B.1.351, which is the South African origin variant, has more mutations. It probably is also more infectious than the wild type virus, but we don't know yet how much more infectious. And there's no evidence, again, that it causes more severe disease. And then we have the P1 variant that originated in Brazil, and that's very similar to the South African variant. What do we know about therapeutics and antibody therapeutics in these variants? The British variant, which is the most common in the US right now, for that, both the Eli Lilly and the Regeneron antibody cocktails still work. These antibodies bind and neutralize the virus. For B1351, the South African variant, Eli Lilly's antibody doesn't bind anymore, and only one of the Regeneron antibodies still neutralizes the virus, the other one doesn't. And we assume the same is true for the Brazilian variant, but we don't have the data for that yet. The South African and Brazilian variant are very rare in the US right now. There's very few isolates that have been identified, while the British variant is really starting to expand quite a bit. What do we know about sera from vaccinated individuals or convalescent sera? It's the same thing. For the British variant, we have very little impact. For the South African variant, there is partially a strong loss, a strong reduction in neutralizing activity from convalescent sera, but also from vaccinated individuals. In some cases, we have seen no reduction in neutralizing activity. In some cases, it was ninefold, so almost a log. And again, for the Brazilian variant, we assume it's similar to the South African variant. What is important to say here is that the mRNA vaccines induce very high titers of neutralizing antibodies. So even if you get a 10-fold reduction, there's still a lot of neutralizing activity left. And for both the Moderna and the Pfizer vaccines, we have seen that protection is already there when you only get one shot, and that's when most people don't have neutralizing antibodies, so very low levels. So it's the assumption here is that with these variants, we'll still see good vaccine efficacy for the RNA vaccines. And of course, there are other mechanisms of immunity, as I mentioned, T cells, memory B cells, non-neutralizing antibodies. So even if the neutralizing activity is impacted, there might still be good protection. And so this is just to sum up what we know about the variants and the vaccines. The J&J vaccine, which hopefully will get licensed in the US within the next few weeks, has a 72% efficacy against the regular SARS-CoV-2 and has some reduction against the South African variant. The same is true for Novavax, where we have 95.6% efficacy against the wild type virus and about 60% against the South African variant. For AstraZeneca, there's data that suggests that there's a loss of efficacy against the South African variant, while this vaccine, of course, still works well against the British variant. And for BioNTech and Moderna, we still don't have data. And I hope that data will be generated at some point. But the assumption here is that it will more look like Novavax and J&J, most likely even better. And we assume that the protection against the South African and the Brazilian variant will still be good. So I think I can skip this. I talked enough about the variants. Just to let you know how many people have been vaccinated worldwide. About 160 million doses have been used. A large proportion of those RNA vaccines. Israel has vaccinated 42% of their population once and 27% twice. So they're really the leaders worldwide in getting people vaccinated. But the US is on a pretty good path too, with 11% of the population vaccinated once and 3.6% vaccinated twice already. So just to conclude, there are several candidates on several vaccines that induce strong neutralizing antibody responses in the non-human primate model and in humans. We see in the non-human primate model that we get good protection against in the lower respiratory tract, good protection against disease, and we see that also in the phase 3 trials. But there is limited protection for the upper respiratory tract, and again, this is also reflected in asymptomatic infections that may still happen in vaccinated individuals. Pfizer and Moderna vaccines are used now in the US under an EUA. J&J is probably the next vaccine that is getting licensed. And Novavax will probably be coming soon as well. And we know now that some variants may reduce vaccine efficacy to some degree, but we do not assume that the vaccines won't work against these variants anymore. And with that, I'll stop, and I'm happy to take any questions.
Wow, thank you very much. That was really amazing. There's a lot of talk right now about trying to give everybody in the country one vaccine first, since there's a limited supply and also limited staffing to deliver the vaccine, would it make sense to sort of give everybody one shot, since we saw in your graph that it confers pretty good immunity, and then maybe delay the second vaccine for a while?
Yeah, so I'm actually I have a strong opinion about that. I don't think that's a good idea. I think people need solid protection. If you have, you know, protection against disease, but you might have already some antibodies, but not enough to prevent, you know, infection or longer term infection. You might actually select for variants that then might escape vaccine responses even better. And also you will see a lot of infections in people who are vaccinated only once, and that will make other people worried about the vaccines. So I think the best strategy is really to try to get people vaccinated twice as soon as possible. And actually that's also what the FDA and the CDC recommend. I wouldn't go the British way, and they are actually basically they are having a longer interval between the prime and the boost. I don't think that's necessarily a good strategy.
Okay, great. There's another question about whether or not people, there's sort of a mythology out there that the more you react to the shot, the greater your immunity is. And especially people who get really severe reactions from the second dose, they're like, yay, that means I'm really immune. Is there any truth to the connection between the reactogenicity and actually people's immunity?
No, not really. I think if you respond with reactogenicity, of course your immune system is working on this, and you probably get a pretty good titer. But there's a lot of people who don't have severe side effects and produce very high antibody titers too. I got a sore arm after the first and the second dose. I never had any other side effects, and I have sky high antibody titers. So there's plenty of people who develop very good antibody titers without having a lot of side effects.
So some of our own doctors have said, oh, we've already seen patients who were COVID positive, they definitely had the disease, and now at months later, they're coming back and they got COVID again. Have there really been cases, and have they been substantiated where people who could get the disease twice?
So yes, there are cases that are documented well. It's very, there are a lot of cases where there's some concern that there might be a reinfection, but very often when you then start to look into that, you don't find enough evidence for that. There are probable cases, and then there's confirmed cases. There's very few confirmed cases because there is very often persistence of RNA. You have to rule out that if your COVID test, the PCR test, is positive, that the disease that occurs the second time is not caused by another infection. So you probably want to run a respiratory panel to exclude that this is now a different bug that does it, but you still have RNA from the initial infection there. The other problem is that in some cases there are persistent infections with people who have a compromised immune system. And so you would actually have to sequence the virus initially and then the second time to show that it's actually a reinfection and not a persistent infection. So once you try to show this scientifically, it's getting very tough, right? But there are cases where it has been documented, and as many things with immune responses and protection, we're talking about risk reduction, not about absolute protection, right? And so it's certainly possible that somebody who had COVID gets it again. It's just very unlikely, but it's not impossible.
So one of the things that people ask a lot of family doctors is, should I take an NSAID or something before the second shot, in order to reduce the reaction and the fever? And does that actually affect the ability for people to respond to the vaccine by making antibody?
So, I mean, I would not recommend that, but a friend of mine who is a B-cell immunologist got the second shot and then got relatively strong side effects. And then he basically took something to alleviate that. And his argument was that once you already have the side effects, the innate immune cells are already at the site, they're already active. And if you take it after the symptoms show up, it shouldn't have an effect on the immune response. But I think he just said that because he felt bad and he needed an excuse. But in general, I don't know. I mean, there are no good guidelines about that. Of course, if you have very severe side effects, you need to do something about that. If it's not tolerable anymore, I would not assume that there is a significant impact on immunity that develops afterwards.
So another question that we're asked is, which of these vaccines, Pfizer or Moderna, is better? People just, just want to know, like, which one should I go get? Which one is it? And is there any difference in the populations? Would you recommend one over the other, or are they both pretty much identical?
They are pretty much identical, also in terms of how they're formulated and what's in there. The Moderna vaccine has more RNA. There are anecdotally there are a little bit more side effects with the Moderna vaccine than with the Pfizer vaccine, but that's anecdotally. There's no good data. The data that we have on antibody titers in a few hundred subjects suggests that they are equal. And their efficacy, although one is 94, the other one is 95, if you look at the confidence intervals, you can't tell them apart. So I would say they work equally well, and there are subtle differences. There are differences in terms of the rate of severe allergic reactions that may occur, but for me, I think they're pretty much equal.
So in the early days, before we knew a lot about the vaccine, there was a lot of talk about the convalescent plasma, and the blood bank in New York was collecting plasma and everything. But it seems to have the enthusiasm for it seems to have fizzled out. Like, it didn't really, work the way people expected, I guess. What's the story with convalescent plasma now?
Yeah, so it's difficult because there are two important factors, right? And we also know that from the monoclonal antibodies. If people are already severely sick, this doesn't work anymore, because the problem that they have is their own immune system and not so much the virus anymore. So you need to give it early. And that's also true for the monoclonal antibody therapeutics. And the other problem is, initially, a lot of plasma was just given without measuring titers and without figuring out how much antibody is actually in there that neutralizes, right? And so I think that makes the interpretation of a lot of initial studies problematic. And then initially, there were also a lot of studies that were not randomized controlled trials. There are data that suggest that the plasma therapy is beneficial if you use high titered plasma and if you use it early. There are, I think, 2 or 3 trials that show that, but there's other trials that show that there is no effect. So it's still a mixed bag. I think it's right now easier to give a more defined product like monoclonal antibodies.
Okay. It says, why is there a lag in data on the mRNA vaccines for the variants? When they were the first to be created, when can we expect real data on these vaccines and their impact on the variants?
So I think Janssen, AstraZeneca, and Novavax decided to have a trial in South Africa, and that was a good decision. Moderna or Pfizer did part of their trial in Brazil, probably for the same reason. You want to have it in different geographic regions, in different populations. But the data in Brazil came out too early to say anything about the Brazilian variant. And keep in mind, these variants just took over in South Africa or in the UK, for example, in late fall, early winter. That's when we already had the results for Moderna and Pfizer. So basically, they would need to now design studies to follow people who got vaccinated with their vaccines in countries that are affected. And they will do that. I mean, we need data on that, but that's basically the reason why there's no data. They were too early for these variants and they were not in the right country.
So there's a question about couple of questions about vaccines in children. So number one, we know that they're often asymptomatic carriers. Or when will we have vaccine? When will there be vaccine for children? And what do you think the recommendations will be about that?
I think Fauci said yesterday that in September we'll have a vaccine, right? So I'm not going to question him. What needs to happen is these phase 3 trials are done in healthy adults, or in some cases in elderly populations. And so if you want to use the vaccines in kids, you have to show that they're safe in kids. You don't necessarily have to show that they're working, but you have to show that they're safe. And Moderna is doing a trial right now to go down to 2 years of age. Usually you do age de-escalation studies, you go down to, let's say, 14 to 18-year-olds. If that looks good, there's no signal, you go down further. The problem that I see a little bit is that children have very strong innate immune systems. And they tend to have stronger reactogenicity. And so if you have a vaccine that's already inducing a lot of reactogenicity in healthy adults, it will be stronger in kids. And that's sometimes or often a problem for the kids, and of course also for the parents. It might still not be dangerous to vaccinate them, but it might be very unpleasant for them. And we had cases, for example, flu, where we had to switch from whole inactivated to split vaccine, and for pertussis, where we went from cellular to acellular, just for those reasons, because they were too reactogenic. But there are vaccines like the Novavax vaccine and maybe the Janssen vaccine coming that have lower reactogenicity and might be more suitable for kids. But we have to wait for the data. It might be that the Moderna vaccine or the Pfizer vaccine work just fine.
So SARS and MERS sort of almost disappeared on their own. How did that happen? And is it possible that one day we're going to wake up and there just won't be any more COVID-19?
Well, SARS disappeared because the people responded very quickly, and it was already in Canada causing nosocomial infections with a case fatality rate of 20%, right? So we were very close to a pandemic in 2003. It could be stopped after 8,000 infections and about 800 deaths, but it was a pretty bad situation. It could probably be stopped because the R0, the reproductive number of the virus, was lower and it wasn't as infectious. There were super spreader events, but in general, it wasn't as infectious as SARS-CoV-2. And usually people were just infectious when they were symptomatic. That's the problem here with SARS-CoV-2. A lot of people, before they develop symptoms, they already spread the virus, and that's hard to control. MERS is a different story. There are MERS cases all the time at low numbers. There was only one larger outbreak with transmission among people, and that was in South Korea. It was also mostly nosocomial transmission. But in general, these viruses are transmitted from camels to humans. The virus sits in the camel population in the Middle East, and if you interact closely with an infected camel, you can get it infected, right? And the camels usually don't have symptoms, or they have flu-like, or I wouldn't say, they basically have common cold like symptoms, at least young camels. And so it certainly can happen that MERS jumps and becomes really transmissible between humans. There's a lot of coronaviruses out there that could potentially do that. There's a lot of flu viruses out there that could potentially do that. And there's a lot of paramyxoviruses out there that could potentially do that. And I don't want to make any kind of doomsday predictions here, but there will be a next pandemic. We had 4 flu pandemics in the last 100 years. Now we have a coronavirus pandemic. There's no reason to believe it will stop. It will come back. Something will come. And we need to get better prepared for that.
So what happened to flu this year? There's no flu.
Yeah, great.
So is it masking that really did it, or did coronavirus just kill all the flu? No, I mean, really, what happened to flu?
It's masking and travel and social distancing. So, viruses have different reproductive numbers, and the reproductive number of flu is about 1.5, 1.6. And it's easy to stop with non-pharmaceutical interventions. And that's exactly what happens. I mean, usually we have, what, 1,700, 1,500 flu cases here at the main campus. This year we had 7. And that's happening worldwide. It's not only happening for flu, it's also happening with human coronaviruses, with adenovirus infections.
Yeah.
Common colds are down, right? And so for flu, there might be 2 consequences. We had a lot of problems with different clades within subtypes, H3, for example, and that made it hard to formulate vaccines. But because the viruses are not going through a bottleneck, we might lose that diversity, and it might make it easier to formulate vaccines again. The other problem, though, is that if you don't have flu for 1 or 2 years, you have now a generation or 1 or 2 years of kids that have never seen flu, and when it comes back, they might amplify it really, really badly, and we might have a really bad flu season. It's not like a pandemic, but might still be a problem when it comes back. So we'll see how that works out.
So do you think going forward, we're just gonna tell people when winter comes to just start masking, and then all of these things will, I mean, if you think about absenteeism and people out of work and everything like that, do you think this could potentially become part of our culture, the masking piece, and that it might be around for a very long time?
So, I mean, of course, like everybody else, I want to go back to normal, and I don't want to have to wear a mask all the time. And I just want to go to a restaurant and, have a nice dinner. But I think in certain circumstances, there's a lot to be learned from Asian countries, right? Where masking is more normal. During wintertime, when you have a flu epidemic, specifically if people sick, they wear masks. And, it might not be a bad idea if it's January and you're using the subway in New York and a lot of people are coughing and sneezing, to actually wear a mask when you use the subway and look a little bit more on your hand hygiene. It's nice to not have a common cold for 1 or 2 years. That's what a lot of people experience now. And I think in a way, if we change our behavior a little, that might make a big impact on common colds and influenza during the winter season, right? We'll see if we learn.
Okay, so I'm gonna ask one more question, and then we'll let you go. What was the ethnic and medical background of those in the study? Was there good representation of multiple populations, African Americans who were healthy and not healthy, and the same with other ethnicities?
Yeah, so that's a very good question, an important question. And actually, yes, Pfizer even, after they already had met their enrollment goal, looked at that and decided that it's not a good representation of the population, and they started to enroll more people from different ethnicities. So in general, these trials are a good representation. And as I said, what usually these companies try is to have multiple locations within a country to also represent geographic spread. And in many cases, they also have sites in different countries, because that also helps to increase the diversity within the clinical trials. So they looked at that. There is no analysis that I know of from these clinical trials in terms of efficacy for specific ethnicities, but in general, a lot of ethnicities were represented similar to their proportion in the US population.
I just want to say thank you. This was really, it's clear. Your presentation was fabulous. Even the answers to these questions coming flying at you, really, 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 really informative. And I know that everybody feels that way. I can just see it. So also record-breaking numbers of people attended. And so just say thank you, thank you, thank you for your effort. And we'd love to have you back in 6 months or a year to hear what's going on as an update. So many thanks.
Thanks for the invitation.
Sure.
Have a nice day.